Thank you for that introduction. I can request the host to share the PowerPoint so that we can go slide by slide. And I think is the screen visible to everybody? Is everybody able to see the screen? Because you have to respond, otherwise online classes like this or online webinars like this will be useless unless until there is a kind of a feedback from you. So whenever there is disconnect or whenever the screen is not visible, you should tell me so that the technical problem can be resolved. So we'll start without any wastage of time. Now the topic that need to be covered in this particular session is all about the forensic molecular biology. So we need to understand how this forensic molecular biology as a specialized subject is little different from the regular molecular biology. So most of us are anyway familiar with what is molecular biology, but then when it comes to forensic science, because the word that is tagged, namely the word forensic, most of us may not be familiar at a student community, because our specialization is not in forensics, or maybe for the first time as a undergraduate student we are exposing to this word, we may not be very familiar with the word forensic. So as the lecture progresses or as the PowerPoint goes on, then we'll understand what forensic is and how is it different from the regular molecular biology. Now the topic is going to cover the among evidences. There are categories of evidences that are presented in the court, before the court. So when we talk of molecular biology, naturally our focus should be on the biological evidences. Now once we say the biological evidences, what constitute the biological evidences? What are the sources of these biological evidences? From where are they coming? When we talk of crime scenes, is it crime scene alone, or is it the accused that we are talking about? Can he be a source of biological evidence? When we talk of victim, can victim alone be source of evidence, or at times as a contamination do we also have to take into consideration the inmates of the house? Because inmates naturally, because they are residing in that house, it is but natural that they also contribute their own biological fluids to that. So how are we going to understand this problem of contamination? Now, why should we focus more on preservation, transportation, and then storage of these biological evidences? What if we are careless about these aspects? What is going to be the consequence of that? That also we try to understand. Now, once we understand the evidence part of it, then comes the laboratory, because there are different phases: pre-laboratory phase, laboratory phase, and then the post-laboratory phase. So the pre-laboratory phase is all about the domain of the investigating officer, because it is the investigating officer, whoever is investigating the case, it is his prerogative, it is his domain, that what are all the methods and methodologies, are the SOPs, standard operating procedures, that he is following, so that at the end of the day he is following the right kind of a methodology, he is following the right way of handling the particular evidence, so that when it reaches the destination, namely the forensic science laboratory, it becomes easy for the expert or the analyst to analyze the pure sample. So as a forensic scientist, you can't expect 100% pure sample, because unlike analytical sample, the forensic samples are subjected to different process, and different factors, external and internal, are going to adversely affect the evidence that is being collected. Am I making sense? Are you able to understand what I am saying? Because I cannot in between talk to you in Hindi. So throughout the PowerPoint I will be using only English as the medium. So if there is any difficulty for anybody at any point of time, you should be vocal, you should be telling me, so that I can go little slow, decrease my pace, etc. Is it okay?
Yes, sir.
So you are able to understand. Now, as we move along, then moving on to the next slide, the DNA analysis, profiling, quantification, there are different stages again. When we process the biological evidences, there are different stages again. Now, in the process, we have the profiling, we have the quantification, we have the interpretation. The screen is not visible to me here. I am not able to see the screen. I don't know for others. Anyway, I have another gadgets I can use. It is visible for me, but I am afraid it may not be visible to the students. Is it visible to the students?
No, ma'am.
I think the coordinator has to take little trouble of again ensuring that the screen is visible to the participants also. Is the screen visible?
No, ma'am.
I think now it is visible. Is it visible to everybody?
Yes, ma'am, now visible.
Now visible. Now, as I said, introduction to forensic molecular biology. Now, when we say there has to be some definition, and why in the first place we are studying the molecular biology is what we need to focus upon. Now, definition, as you know, any forensic, when you add the word, tag the word forensic, it only means that this particular technique or technology is going to be used on a matter or on an issue which is there before the court. Before the court. Matters become issues when there are two parties for it, and then one party claiming the right, the other party saying that we have not done harm to the right. So one party for the issue, the other party naturally will be against the issue. So there are two parties, and whenever there is a dispute, there ought to be two parties, and that particular problem, if it is legal, has to be before the court, and it is the court, or rather the presiding officer, who has to resolve the issues. So how many of you in the webinar group are familiarized with the word forensic? How many of you are, can anybody in the class tell me what the word forensic exactly means? Is there anybody who can tell me? Yes? There are 27 participants. I wish you should be vocal. It may be a wrong answer, just don't bother. If you are interactive, then you will learn more. So any one of the participants among the 27 should be able to tell me what exactly is the meaning of the word forensic, and why should we learn forensics in the first place. You have subjects like forensic biology, forensic chemistry, you have subjects in the syllabus? So somebody should venture, somebody should be bold enough, even if it means it's a wrong answer, doesn't matter, but you have to tell me what is the meaning of the word forensic. Anybody, any guesses? No? No problem. But as I said, whenever there is a legal matter which becomes an issue, then there are two parties for it, and in legal parlance we call one group as the prosecution, the other group as the cross-examination or the defense. So prosecution, defense are the two agencies before the court. One party, namely the prosecution, representing the victim, because victim means, because the crime is a subject matter of the state, and victim will be the accused will be represented because it is the right of the society. It is not only right of the victim, it is right of the society at large. Hence, for that matter, accused is always represented by the state, and it will be, if you see the citation of any court, the accused versus will be the state, versus the state will be the citation. It simply means that the accused is represented by the state, and you see the name of the victim. Now, If that is the case, now in the court, whether it is the investigating officer or whether it is the forensic scientist or whether it is a prosecuting agency, whatever be the agency in the criminal justice system, because we are all part of stakeholders of the criminal justice system. Now, if that being the case, now the matters which are before the court, the matters can be in the form of evidence, and evidences are, they can take the form of a biological evidence, they can be physical evidences, they can be documentary evidences, or there may be a direct eyewitness for the case who happened to be there and who witnessed the case happening before his very eyes. So that way he becomes the eyewitness. Now, if that is the case, now whatever knowledge the expert has got in that particular field, let us say a molecular biologist is the expert there. Now, why is he called to the court? Now, he is going to analyze whatever evidences that have been collected by the investigating agencies in the scene of crime in the form of biological evidences, for example, blood, semen, saliva, or whatever is the nature of the biological material. The investigating officer, as the crime is reported, rushes to the scene of offense, and then there he collects all the evidences, and then these evidences he will be forwarding it to the forensic science laboratory. Now, simply forwarding doesn't help. Now he has to make sure that whatever he is collecting is collected in a proper way, following a particular procedure, and then once he ensures that it is free of contamination, cross contamination, and there is a proper custody. So there are a set of custodians. Authorized custodians are the only ones who are responsible for the travel of the physical evidence. Now, the investigating officer, once he collects these evidences by using proper SOPs and then proper procedures, he will be sending the same to the lab with a request. There is a proper documentation also. So what is that documentation? Documentation is talking about what is the type of evidence that he has collected, from where he has collected, what is the methodology he has adopted, and what kind of package he has put the evidence into, and then what type of information he is trying to seek from the expert is what is written in the document. And once the moment the evidences reach the forensic science laboratory, now the actual examination or the analysis is going to start. Now, not immediately. So there is again everywhere, when you talk of court, every step in the travel of the evidence right from the scene to the court, there has to be a proper custody, proper maintenance of the evidence. Now, when he takes up in the laboratory, the staff who open the case and then ensure whatever is written in the document, is it in its proper shape? Has it been received in the physical form also? Is the evidence that is mentioned in the letter, is it the same that is been received in the physical format? So after ensuring all that, after documenting all that, the evidence reaches the proper scientist. That means when we talk of biology, namely the blood and other evidences, if they are there in a particular crime or a case, they will be referred to the biology section of the forensic science laboratory. Now, we are going to, there is molecular biology is a set of instruction or a specialized science where you are going to study the molecules which are biological in nature. Then it can be a macromolecule like DNA, it can be a macromolecule like RNA, or it can be a protein, or it can be enzyme, or it can be anything, as long as it pertains to its nature is biological in nature. Now, so that particular knowledge the scientist has got as molecular biology, he is going to apply that particular knowledge onto the case, onto the issue which is before the court. Now he does a battery of examinations, a series of analysis, and then after the set of examinations get over on that particular evidence, he is going to formulate what is known as his opinion. So the oral opinion doesn't work. The opinion which has been reduced into writing is otherwise called as the report, forensic report, or some reports are also called as opinions. Like for example, document somebody is going to give a report, it will not be called as a report, but rather it will be called as an opinion of a document expert. Now, in the Evidence Act also, the definition for the expert, whether he is a biological expert or a document expert or a fingerprint expert, the definition for the expert is already given in the Evidence Act, and also the same thing gets reflected in the Naya Kanun also. Naya Kanun is not entirely a different or entirely new thing, only some sections and then some chapters, they are put a little forward, backward. So only that has happened, but not much drastic change has never happened in the Naya Kanun also. If you take all the three major acts also, nothing, sea change has not taken place. Now, so as per the definition of the expert, you should have the qualification, you should have the experience, you should have the expertise in this particular field. Now, all that experience, all that knowledge he is going to apply, he is going to generate, he is going to perform the analysis, he is going to generate the reports, and once the reports are ready, now again the evidences need to be packed in a proper way. As he has received from the investigating officer in a similar fashion, he has to properly do the packing, repacking again, and then produce them before the court of law. So this is precisely is the travel of any evidence. It need not be biological evidence, but it will be by and large the travel of the evidence right from the point from where they have been located to the point where they need to be submitted. So finally they will be submitted in the court of law. Now if you see the definition, now the application of molecular biology techniques, what do you understand? We have some knowledge, and that particular knowledge we are straight away trying to apply the technique to the forensic, in the forensic side. That means what? So what is the issue? There is a murder that has taken place. Now we have found some blood in the scene, in the crime scene. We have no idea from where this particular blood has originated. Is it the blood of the accused, or is it the blood of the victim, or maybe somebody else, some inmate got injured in the process? We have absolutely no idea. So if it is copious blood, definitely some guesses can be made that it could be a victim's blood. But maybe in the struggle, the accused might also get injured, and that's how you also see the accused blood. Or accused may be not one, there may be 2, 3 or multiple people, or inmates may be there, or some passerby may be there. So there are so many people, the possibility that other person's blood other than the victim and accused may also be there. So by looking at the red color of the blood, nobody will be in a position, including the expert, will not be in a position to tell straight away, yes, this blood is somebody else's and this blood belongs to somebody, some person XYZ. So it is very difficult unless you perform a scientific test over there and then do the analysis. You will not be in a position to tell whose blood it is. Now for this sake, it is mandatory that whatever knowledge you have gained, you are going to apply that and then do the analysis in the lab and then say, yes, this is the blood of a human and this is the person from whom it has come. So not only the study or the knowledge in the biology, the knowledge in serology, knowledge in DNA, that has to be applied. From all the fields, the knowledge has to be applied. And since it is only application of knowledge, so whenever we apply pure science knowledge to matters that are before the court, we call that particular science as the forensic science. So the word forensic has got a root from the word forum. Forum, malls ka bhi forum bolte na, where public gather for justice. So forum is the word, or forensis is the word from where the forensic science got the root. Now forensis or forum means it is a public place where people gather for the justice. This is simply the meaning of that. Now what is the importance? Why do we study forensic science? Now we have to study forensic science because there are crucial matters before the court on which the trier of the fact, namely the court, doesn't have knowledge. You can't expect court or the judge to have knowledge of every subject. That is the reason the areas in which he has no knowledge, he is employing some people, some experts, so that the expert with his knowledge can help the finder of the fact with the facts and figures, so that he can come to a logical conclusion, because he is unaware, he is not, he has no knowledge, domain knowledge. So that is the reason in certain areas, as I have already mentioned, under the definition of old Act 45, Indian Evidence Act, a list of experts is already given. So those experts' evidence will be accepted, will be admitted in the court of law, subjected to certain criteria. So since it's not a law class, I am not going to deal much on the Evidence Act or the admissibility factor, but this is a very small degree or undergraduation program, so I don't venture to take you more into law. So that becomes little complicated for somebody to understand or comprehend. Now that we have understood the importance. So everywhere forensic science is finding its place. You name accounting, forensic accounting is there. You name engineering, forensic engineering is there. Isn't it? You say nanotechnology, forensic nanotechnology is there. Isn't it? You mentioned dentistry, forensic dentistry is there. So every walk of life, forensic is entering. Now it has become so popular, and with Naya Kanun also lot of importance and significance has been thrown, and it has become mandatory. The law provides, that act mandates that a case where the crime attracts the punishment of 7 years or more, they said it is mandatory for the forensic scientist to accompany the investigating officer. That is the reason in all undergraduate programs, more and more colleges are going to introduce the subject forensic science. I think I am making it quite clear to bring it to the knowledge of the students that why in the first place we have to study forensic science, and why even at the undergraduation level some colleges are keen to introduce this introduce forensic science as a part of the curriculum. So can I move on to the second slide?
Yes, ma'am.
The organizer should move to the second slide. Now the biological evidence collection. When we say biological evidence collection, we have to know what is the definition of that, and how many types of biological evidence that one can encounter in the scenes of offense, and what should be the method. See, so when we talk of method, I'll tell you in more details why we have to adopt certain methods. Now, definition part of it, when we say collection, what do we mean by collection? Gathering biological material, because in the crime scene, biological material or matters are scattered everywhere. It may be in the kitchen, it may be in the bathroom, or it may be in the living room. So wherever this scuffle or wherever the interaction happens between the accused and the victim, in all such places you are going to find materials, biological material. Maybe the accused, out of tension, out of stress, he must have smoked there. So he is leaving his cigarette butts there, cigarette stubs there. So they may contain his own saliva. He might have drank from the glass a cup of coffee or teacup. So in that he is going to have his lip prints. So wherever possible, depending upon the type of crime that has happened, you are bound to get all the biological evidence. But it is mandatory on the part of the investigating officer. He should have a thorough knowledge that what kind of evidences can be there in what kind of crimes. Say murder, what probable evidences can somebody find, and where exactly can somebody look for that type of evidences. So gathering all kinds of material objects which are biological in nature from a crime scene is the first and foremost job of the investigating officer. Now, once we understand the definition, what is the biological evidence? Now, moving on to the types of biological evidences. So there are n number of biological evidences. When you talk of physiological fluids or biological, then a previous slide, move on to the previous slide. I have not completed the previous slide. See, when we talk of biological evidences, not only the biological fluids or physiological fluids, we understand that blood, saliva, semen, then amniotic fluid or the synovial fluid. There are so many biological or physiological fluids, and apart from that we may encounter hair, we may encounter skin, we may encounter bone mass, or we may encounter some plant material, a cannabis or some narcotic drug, in which we are interested to know whether it is a narcotic drug or a plant material which has got narcotic origin. So all these, and not only plant material, when we talk of biological evidence, we have to include even the animal material. For example, the wildlife crime is reported where a particular tissue belonging to a particular animal is found in the scene of offence. We have already heard about Salman's case of black buck. So, in that there are, as per the Act, Wildlife Prevention Act, there are a number of acts. One such act talks about the preservation of the wildlife. Now, in order to cut the balance, we have talked about the act. Now, in that, there are the animals which are listed as endangered, which are listed as extinct. So, if that material belonging to that particular scheduled item, scheduled animal, then if you can prove by biological analysis that this sample belong to an animal which is already enlisted or which is already put in the schedule, then he is going to, and once you also identify the accused, then that particular accused will be punished under this section because he is in possession, or he has killed, or he has hunted, or he is in possession of that particular animal material. So, biological evidence include a gamut of evidences, not only plant, not only animal, not only human, and not only insects. Insects also are there. For example, there is a dead body, and the time since death of that body is not known. Now, how do you go about it? Now, the only way that you go about it is collect all the insects or the insect population that is dwelling on the dead body, and then send it to the lab and then ask what is the species and how long the species has been dwelling on the dead body, so that you can calculate the life cycle of that particular insect and then back calculate and then try to establish the times. So this part of forensic is called forensic entomology. So forensic is pervading in every area. Now, so when we talk of biological evidence, all these things will come into picture. Now, what are the methods? So why should we talk about methods? Now, every time it is not possible for us to for example, let us say there is a blood stain, there is a spatter of blood on the wall, let us say in the crime scene. Now you cannot lift the entire wall and then send it across. Humanly not possible. We are not Hanumans. It is not possible for us to do that. So, in order to facilitate easy movement or easy transport of the biological evidence, we resort to lot of methods of collection. One such method is swabbing. Now, if swabbing is nothing, when you go to a doctor for some diagnosis, what does he do? He tries to give you an injection, intramuscular injection or intravenous. Before that, what he does, he dips a roll of cotton in some spirit or in some disinfectant, and he is going to rub it across the skin. That process of rubbing is called swabbing. Now, swabbing, now it may be the surface is such that swabbing may not be possible for us. So what is the alternative left? Now it is scraping. You scrape the surface nicely with a knife or with a blunt edged thing, so that the layer, the outer layer nicely which contain the blood can come out. So that process is called scraping. Now it again depends which method you are going to use will again be dependent on the surface on which the material is present. For example, it may be the blood, if it is there on the table, then you can go for scraping. If it is there on a wall, you can go for swabbing. So like that, or if it is possible to dismantle that particular substratum and take out only that sample, if that is feasible, then you can. It's all commonsensical. So the method, the appropriate method has to be used. So what will happen if a wrong method is used? why should we talk of good methods and proper methods, right methods? What if somebody adopts or if somebody erroneously does some method, what is the consequence? Now, as such, the forensic samples, you can, unlike analytical samples, you are not going to get it again and again. Once you lost it, it will be lost forever. Once you lose it, you are losing it forever. You will not be in a position to regenerate it. It is permanently lost, isn't it? That is the reason why whenever you collect these samples from the crime scene, you have to be so meticulous so that you are using the right method and right handling. Only that is going to give you the effective end results. What if you are going to use a wrong method? What if you are going to use a wrong method and then use a very right kind of a handling? Even that is also is going to be disastrous. So any combination, other permutation other than the right handling and right methodology use, all other permutation combinations are going to be futile. So that is the reason why lot of importance or significance is being given to the method that has to be adopted in order to take out the material, the incriminating material that is available from the scene of offense to the science laboratory. So it can be a swabbing method, it can be a scraping method, it can be a casting method. So depending upon on what surface you are going to get the biological material, that appropriate method of collection has to be used. So these methods, whenever the incriminating material is non-portable, that means it cannot be transported easily, then we resort to these methods. Otherwise, if it is a portable article, it is there, the blood stain is there on a paperweight, very small, portable, easy to carry. So the best method is, what you need to do is, you have to dry that particular, because most of the time the blood is still wet. So you are not supposed to send a wet sample, because wet samples are already prone to, always are prone to the degradation by the bacterial action or microbial action. So it is worthwhile that whenever the blood sample is wet, then you are supposed to dry it under shade, under shade, and then ensure that it is not dried under the sun. Then the reason is why. So whenever we say, we have to question ourselves, why this method, why not the other method? Now, that way if you probe into this Aristotle type of questioning, then you will remember that particular principle for a very long time. So when we say, why not in sunshine or why not under sun? Now, the reason is that these materials being biological, the proteins, the enzymes or the molecules that we are going to test in the blood, they may get lost, because they may be thermolabile, they may get destroyed under heat. So that is the reason why we say that the material has to be, when it is biological in nature, and that too when it is blood, has to be properly dried under shade, because the moment you put the material in sun, it is going to lose, it is going to degrade, it is going to decompose. Now we also say, once it is properly dried, then the next thing is that you have to, whenever you talk of biological material, then you have to use the package called porous material. That means porous material, it can be a paper, brown paper, white paper, cardboard box, or it can be a paper bag, it can be a cloth, it can be anything. Now the reason why we use porous material for such biological evidences is very simple, that if there is no aeration, if the package is going to be airtight, it doesn't allow air to go from inside or allow outside air to enter inside. Now the result is, if the biological material is put in a airtight packaging, now it is creating what is known as anaerobic condition, that means no air at all. Now whenever there is no air and whenever there is a biological material present, now lot of bacterial activity happens there. Anaerobic bacteria are going to interact, and then once they start interacting with the biological material, they are going to chemically modify that, they are going to make it so vulnerable that after their action nothing is left. Whatever the material that you intend to look at in the lab, is going to completely degrade, completely destroyed, with the result you may be present thinking that lot of blood is there, I am going to get some result out of it, but then when actually it goes to the lab, when the expert does the proper analysis, to his surprise he is not going to get any result. So nothing is detected. The reason is the material to detect blood is not at all present in there. So each days of delay, so 0 day, as the moment it is been located, you have not wasted any single minute and you have immediately transported. The evidentiary value of that particular thing is going to be 100% at the end of the day. Now each day's delay, if you go on keeping on delaying, so first day you are preoccupied, you are so busy, you are not able to send it across. Day one, some material, some evidentiary value is lost. And let us say after 1 month, you have taken so long that 1 month has already elapsed, and after 1 month if you are going to submit the material to the lab, now you may be thinking that there is copious blood present over there and scientist is definitely is finding out some truth out of it, but to the surprise of everybody, now on 30th day, if you see the evidentiary value, because you are not going to get anything out of that. So what will be the evidentiary value at the end of the 30th day or at the end of the month? Then it is going to be 0. So this is the significance that whenever biological evidence is encountered in the scenes, no time delay is excusable. No time delay is excusable. And of course, court is not going to vehemently say that there is a delay, we will not give any conviction, rather we are going to acquit. But the reasons have to be justified why there is delay. If the delay is justifiable, or if it is substantiated, or if the IO is going to give the proper reasons. If suppose the crime is not reported at all, then what is the police officer is going to do? What the scientist is going to do? If the lapse is on the part of the victim only, then it is excusable. But then a deliberate, wanton kind of a delay on the part of the investigating officer without justification is not at all tolerable, and it becomes vexatious to the case. So that is the reason the trier of the fact will become helpless. He will not be able to render any kind of a verdict on that. So many things are related to the delay. So that is the reason why delays have to be avoided. Moving on to the next slide. Please move to this next slide. Now, we have to talk about the sources of biological evidence, as I have already told you. So biological evidences can take different forms. It can be blood, it can be semen. When you talk of rape cases or molestation or POCSO cases or unnatural offence, whatever be the nature of the crime that you are dealing, then these evidences, the semen of the accused, which will prove his presence. So whenever we talk of biological evidence, we have to talk of 3 evidences. One is proof of presence. So what do you understand proof of presence? That particular person is present in the scene of offence is what is proof of presence. Proof of association. That means this accused has got an association with the victim. Now, proof of presence, proof of association, and proof of contact. That this is the person who got contact with the victim, he is depositing his semen on the private parts of the lady and all. So these 3 proofs can be established whenever you find the semen on the victim's of the garment or on her private parts or whatever be the source, they will be present. Now you talk of saliva. Saliva also becomes very, very important in sexual assault cases, in murder cases. Whenever you find these things, they will establish the presence of the accused. Now urine may be there, hair samples, as I have already discussed, rape cases, or some weapon is, a murder weapon is used by the accused. Now the murder weapon, while it is employed on the head of the victim, it is going to collect along with the blood some hair samples, head hair samples of the victim also. Now if it is collected from the possession of the accused, then it will have blood, it will have the fingerprint of the accused, it will also have the hair sample of the victim, blood of the victim. So if you can establish in the lab that this hair is that of victim, this blood is that of victim, and this fingerprint is that of accused. If you can establish these things by way of analysis, then you are identifying the perpetrator. That means the person who has done the crime. Now, teeth also are most frequently, whenever suppose there is a mass disaster, man-made or naturally occurring, then or it is an aeroplane crash, then at the end of the few days, maybe the identification is not yet established. All that you find in the disaster is only teeth sample. Now teeth samples are also important. Now the ante-mortem record, that means the records which are there existing before the death of the person, of which there is a record. If you compare the ante-mortem records with the post-mortem records, you can establish the identity of the person through teeth. Now bones also likewise. So in fire accidents, in other accidents where no tissue is left except for the bone, then bone is the only source of biological evidence that we find in the scenes. Like that, any soft tissue, muscle tissue, skin tissue, it can be any type of skin tissue. Now what is very important for us is, we require only very small amount of blood when we talk of DNA. But all other tests like presumptive tests, confirmatory tests, or the ABO identification, or the secretar status, or for that matter any other enzyme patterns and all may require more sample. So that is the reason why we have, because forensic samples, the scientist or the investigating officer doesn't have the luxury of having the sample in a very large quantity. That is one problem, and we are also not very sure whether the sample is a pure sample or it is a mixture of samples. We are also are not aware, and so that is the reason why the old traditional tests we are not able to decipher anything. Now we have, that is the reason why we have shifted to more and more sophisticated technologies, and one such technology is our DNA technologies, where we don't require blood in large quantity. All that we require is only a speck of blood, or a single strand of a hair, or one bone piece. So these are the only things which we are going to, at times in some cases these are the only materials available. So in such cases, this cutting edge technologies like DNA, where even small quantities also we can analyze that sample, we can identify the people. So that is the reason why more and more technologies are going to be embraced by the forensic scientists, so that it becomes easy for us to analyze and then come to some logical conclusions. Moving on to the other slide. Please change the slide. Now, why should we bother about knowing the best practices for evidence collection? As I told you already, now if best practices not been followed while you collect, while you pack, while you lift, and while you transport, and while you store these evidences, now what is going to happen? It is going to adversely affect the analysis of that particular material. So that is the reason why wherever SOPs, standard operating procedures, are prescribed, wherever best practices are given in the form of a textbook or in the form of a manual, it is but natural that we have to adopt those best practices, so that whatever we are collecting, it is in its proper shape and size. Now, what are all the best practices that somebody has to master in order to ensure that when the evidence reaches or when the material reaches the lab, it is intact? Now, one such a good practice is wear gloves. That means whenever you handle the evidences, the best practice is you have to wear gloves. So what is the advantage of wearing gloves? Now, for example, you have to handle, let us say, a blood-stained knife. Now, blood-stained knife may also have fingerprints, because latent fingerprints are not visible to our eye, naked eye. They are not visible at all. A sweaty print is not visible to us. So without visibility, if you are handling, what will happen? You are not only destroying the evidence in the form of a fingerprint of the accused, and if you don't wear gloves, what will happen? You also, because you are also sweating, it is also a summer day, you are also sweating, your own fingerprint may be present over there. So you are doing a double harm there. One is, you are already destroying by not wearing gloves, you are destroying the fingerprint of the accused, which is already there. Number two damage is that by not wearing gloves, you are also spoiling you are also adding your own fingerprint over there. So, to avoid this kind of a contamination, cross contamination, it is good practice always to wear the gloves. Now, use sterile tools. Why should we? I told you that for DNA technologies, we don't require much blood. It is so sensitive a technique that you don't require blood in quantities. So, that means what? Even if a small biological material other than the incriminating material is present, in the test you are going to get positive for that also. So I'll give you a small example. Now you are using, the tool that you are using is not sterile, for example, let us say. Now, which is a contaminated tool. Now if you use that, what will happen? Now for the first usage, you have already taken one sample, and the same tool again and again if you are using, you are adding contamination to it. So it is but natural that whenever you use tools, it should be for a single use as far as possible. When you talk of DNA, you have to be very, very meticulous in collection methodologies. Now, first one, you have to use a sterile one, and then the same tool should not be used again and again. Use and throw. Once you use, it cannot be used for the other thing. So such type of tools are also coming up commercially also. You have to use such sterile tools. Now, document everything. So I told you already, the court, eyes and ears are only documents, because court is going to, judge is going to come and visit every crime scene, isn't it? Now, it is only through the document. So whatever we do, whatever we perform, either be the forensic scientist or whether it is the investigating officer, whatever activity that they do in the crime scenes or whatever activity the forensic scientist does in the lab, so they have to properly document, record everything at one place. The recording should contain the observations. Yes? There is some cross talk there. Is somebody not clear about it, or is the disconnectivity? Yes? Since all the participants should be on mute, otherwise there is a lot of cross talking over there. I don't know whether I am audible or whether somebody is following it or not. There is no absolute feedback from your side. There is some cross talking. The organizer should unmute everybody, mute everybody, because there is a lot of cross talking happening. Yes, should we continue, go across?
Yes, ma'am.
So let there not be cross talking, because somebody is not clear, they can unmute themselves and then they can tell their difficulty instead of talking among themselves. Am I clear so far?
Yes, ma'am.
So let's move on. Now document, you have understood what is the significance of documenting everything that is there in the scene of offense. Now, document simply means that whatever activity that we perform, whatever job that we do in the scene of offense, the IO has to write it either in CD. CD means case diary part 1, case diary part. There are a lot of documents that have to be maintained by the IO. He is going to scrupulously, meticulously going to write all that in the document. Now, document everything. That means record the location and condition of the, from where he has collected, who has collected, who has lifted, who has packed, who has written the document, and all, everything has to be properly documented. Now, these are some of the best practices. Moving on to the preservation part of the biological evidence, we'll move to the other slide, which talks about the preservation of biological evidence. Move to the second slide, next slide. Huh? You are not at all audible. I am not able to hear anything. Yes? Should we move on? Yes? Yes, that's what we are talking about. See, preservation of biological evidence, why preservation is again important. What happens if we don't preserve? So, what do we understand? What is the definition of preserving the biology? Maintaining the integrity of the evidence after collection. That is the definition that is being given. Now, let us understand what do we mean by the integrity of the evidence. For example, let us say there is a blood stain present over there, and what I have done, or there is a blood stained garment of the victim, let us say. So the victim is murdered and the garments of the victim have to be collected, and the investigating officer goes to the scene. He is going to collect the victim's blood stained cloth, let us say. Now in the process of collecting, because it happens to be a very sunny day, and the investigating officer is sweating all over, and then inadvertently, without knowing that his sweat also can contain some the blood grouping substances, so he is touching the blood stained garment all over, and then he dries it, and then he sends it across to the lab, thinking that he has done the proper way of handling and proper way of sending the material object to the scene. And after it reaches the lab, the concerned scientist or concerned analyst is going to analyze, and then he is getting two groups. Can there be two groups for a single person? Can there be two kinds of blood grouping for an individual? Or it has to be only one group? Can a person have A, O, B, all groups, or he has to either have A group, AB group, O group, like that? So all possible combinations will be there, permutation combination, or an individual will have only one blood group. Is my question clear?
Only one.
That means if every person can have every other group, there is no meaning. We will not consider that as evidence, isn't it? We will not go to perform that particular test where we are ambiguous, everybody is getting every group. But surprisingly, if a contaminated exhibit is sent to the lab, what will happen? Now, you may get A group on account of the victim, and because the person who handled that evidence, because his sweat is also being added as contaminant, because he is not wearing any gloves. So when that is being added as a, because most of the population, we are all secretors, that means we secrete the same blood group even in our body fluids. Other than the blood also, these people secrete the blood grouping substances in their body, other body fluids also. So let us presume that this person is also a secretor. Through his sweat, he is adding all his blood grouping substances to the, let us say, to the garment. So that is the reason why we are also getting B group. So A group is there, B group is there. So we cannot just say that he is AB group. It may be A and B separate. So all these kind of cross contaminations will be there, and integrity of the evidence is being lost. Integrity, its integrity is lost. You cannot consider that as an authentic sample. Integrity is lost. So it is always the defense who try to prove that there is no proper integrity of the evidence that is maintained. So it is one of the pet questions of the defense to shatter the credibility of the expert, or they shatter the credibility of the IO. They will say, see sir, there is no record to prove that this evidence integrity is properly maintained at the time of collection. So he will raise this question, he will raise this doubt in the mind of the court. So that is the reason why when you talk of biological evidences, a proper preservation has to, that means a proper preservative. For example, we have drawn the blood sample in a paternity case, we have drawn the blood sample of the father, putative father, mother and the child, because there is a dispute in the parentage. So in drawing blood, suppose if you don't add any preservative, what will happen? Blood without preservative, when it is reaching the lab, what is the consequence of that? Is it going to be intact? Yes, what will happen to what is the fate of the blood sample which reaches the lab without any preservative? What is the fate of it? Yes, anybody?
Maybe it would be not, or it would be discussed.
So the consequence is so bad that it is completely destroyed. Now when you are sending a destroyed, damaged, disintegrated, degraded sample to the lab, you cannot expect some wonder to happen in the lab, isn't it? So garbage in, what is the result? Garbage in, what comes out? Gold comes out? What comes out? If garbage is in, what is the outcome? If you put garbage, are you going to get gold out of it? What is the outcome? So GIGO principle, garbage in, garbage out is the only outcome. You cannot expect some wonder to happen, isn't it? So that is the significance of the definition of maintaining the integrity of the evidence after the collection. Now what are all the techniques that we have to, not only using the preservative, right kind of a preservative, and then you have to dry it properly. When I talked about the blood stain garment, I said air dried. It has to be air dried, that it has to be dried under the fan, under the shade. Now if you are going to dry it under the sun, it is going to lose its property. So it has to be proper freezing, refrigeration, or other preservatives have to be used in order to ensure that you have ensured the integrity of the evidence when it reaches the forensic science laboratory. Now, moving on to the other slide, which talks about the transportation of biological evidence. Please move to the other slide. Yes. So this particular slide is talking about the transportation of biological evidence. So why should we transport? Now, crime is happening at one place, and analysis is going to happen at some other place, isn't it? So for that reason, the biological material has to be transported from one area to the other. Now in the process, so there has to be some container, there has to be some format, there has to be some proper package and all. So this process of using all these will be, in consolidated, it is called as transportation of biological evidence. Now what is that you have to use? You may have to use some containers. So when you talk of biological materials, I told you it has to be the packaging material has to be used. That has to be used for transportation, has to be porous. It should allow air in and air out. So such materials can be cardboard boxes, small cardboard boxes, they can be paper bags, they can be bags made of cloth. So it has to be, the only criteria that has to be fulfilled is called the porous material, and of course it has to be sterile. The container itself should not be a one which is already dirty, which has already been contaminated. So you should avoid such containers. So the two criteria that are required for transporting the biological material is, one is it has to be porous, the other one is a sterile container has to be, and the container has to fit the material object, isn't it? So it should not be too small, it should not be too big, isn't it? It should be a proper appropriate kind of a container that has to be used. Now, what do you understand by labels? Labels clearly label with relevant information. So what do you understand? Now, it is the IO who is, the investigating officer, who is the one who is collecting the material, isn't it? He knows from where he has collected. He knows from which corner of the crime scene he has collected. But then if he doesn't communicate the same to the person in the lab, how will he understand? So for that purpose, each exhibit that you collect, it should bear what is known as the label. So the minimum information that should go to make a label is what is the name of that exhibit, from where he has collected, how he has collected, on which date he has collected, what is the number. Suppose there are 2-3 evidence material. So what is the number, serial number that is given to that particular exhibit. So when and what date, at what time this particular material object is been collected, and who is the one who is collecting it, and then what is the name of the investigating officer. And the most important thing from the legal point of view is, whenever an activity in the crime scene happens, the investigating officer should ensure that 2 independent witnesses from the locality, from the nearby place, have to be there. So all the activities should happen in their presence. They are called Caesar punch witnesses. So this has to be done in their presence. Not only that, how the court is going to believe that you have done this in the presence of witnesses? Only by their signatures. So that label should also contain the signature of the witnesses. Not only sufficient, but then the IO, the investigating officer who is investigating that case, should also append his signature. So that means this is the minimum information that the label should contain, and such label should be there on all the exhibits which you are going to collect from the scene of offence. I hope I am quite clear about the label, the significance of the label, the contents that should be, the content which should be there in the label. I hope I am clear. Am I clear?
Yes, ma'am.
Ha. So once we understand the label, then we need to move on to what is known as very, very important when you talk of forensic science, or when you talk of matters before the court. Now it is chain of custody. Now I told you that there are so many stakeholders, there are so many agencies that are involved in the travel of the evidence, right from the site where it is been located to the place where they are finally deposited for the trial. Isn't it? There are so many agencies. First agency is first responder. Who will be the first responders? Who have located the evidence in the scene of offence? What is that agency? Which agency will be the first responder who will go to the scene of offence as and when the crime is reported?
Forensic reporters.
Forensic reporters, these days they are going. Earlier it is only the police officer, isn't it? Now because of the Naya Kanun, even we are also, forensic scientists are also going. But then earlier days it is the only police officer, not the police officer of a higher rank, it is the constable. For that matter, constable is the one who is going to go there. So for that, let us say the police person has gone there. Now after collecting all the evidences from the scene, the police officer, to whom is he going to hand over? Which agency comes into picture next? Which agency comes into picture next after police? To whom it will be? Yes? Forensic science laboratory. Yes, good. The forensic science laboratory is going to hand over. That is the second agency. Now, how do the court understand? It is okay physically happening, but when it comes to court, it will take several years. So how can somebody remember, yes, I have given on that particular day this evidence, this box to the lab? Who will say? By what means? Court has to be convinced, isn't it? So there needs to be a proper custody, of chain of custody. Who are all the authority who has handled these evidences, and from which place to which place it has travelled, and what is the name of that person? Is he an authorized person, unauthorized person? Does it get reflected in the document and all? So the entire thing is produced in the form of a chain of custody. So this is called chain of custody. Who is handing it to whom? Who is the agency? Who is handing it to whom? Which agency? Next agency is reflected in the chain of custody. So you have to maintain. The prosecuting agency always has to, the burden is on them to show that a proper custody, chain of custody, a proper integrity of the evidence is maintained throughout the travel of the physical evidence, is what is to be established in the court of law, and the burden is squarely on the prosecution to do that. So we have understood the concept of integrity, we have understood the concept of chain of custody. Now moving on to the other aspect that is called the storage of biological evidences. Let us move on to the other slide to understand that. Now, storage of biological evidence. What do you know? What do you understand by that? Why should we store the biological evidence in a proper manner? Now, there are several factors which are ready to damage the biological evidence. Some of them we have already discussed. One is environment. So, you cannot control the environment. Environment as per the seasons will keep on changing. Now, what is there in your hand is, according to the environment that is prevailing, what best methods can you use in order to ensure that this environment doesn't have any adverse effect on the material that we are handling. Now, store in appropriate conditions. Suppose a material has to be preserved in the freeze. So ensure that there is freezing there. You have to ensure that it has to be dried. Ensure that it is dried. So like that, in order to prevent the environmental adverse effect on the material, you have to adopt the storage processes for the biological material. Now temperature is another thing. I told you the biological materials, when you expose them to heat and all, adverse heat or very high temperatures, then the material is going to adversely affect. So where it has to be kept in cold, where it has to be frozen, so you have to adopt those methods of storage and preservation, so that when the material reaches the forensic science laboratory, the person who is doing the analysis will be in a comfortable position to do the analysis, and then at the end get some some results which are useful for the court. Now documentation is another way of storing. Now documentation is very, very keen. I told you already that whatever activity that you perform, either in the field or in the lab, that has to be reflected. So only through this the court is going to examine the activity. Otherwise you can't expect physically the judge coming to every scene and observing what you are doing, whether you are doing it correctly or whether you are doing a wrong procedure and all. He cannot do that, humanly not possible. So your document is your mouth. Your document should speak for your activity. So that is the area where you have to be very, very critical, where you have to be very, very keen to maintain a proper documentation. Now moving on to the other slide, which will talk about the multidisciplinary approach of the work in the forensics. Why should there be a multi-agency or multidiscipline that is involved? Now as I told you, so lot of collaboration work has to take place. Now when we talk of a disaster management, for example, disaster management, because the bodies have gone beyond recognition, you are not able to identify the body by its physical feature. So then you have to call an anthropologist there, you may have to call a dentist there, you may have to call an osteo specialist in bones, or you may have to call a forensic medicine doctor there. So you may have to collaborate and involve lot of agencies. Hence we say that forensic work is always a multidisciplinary kind of a approach. Now there may be legal experts. So when you give the report, the legal expert has to ensure, the prosecutor has to ensure that whether the crime has been defined properly, whether the proper section of law has been included, whether it is a 302 case or 375 case, what kind of section is getting attracted. So legal experts are also important, and scientists, whether it is blood or whether it is animal blood or whether it is hair and all. One second. So all that is very important. Now that is the reason why it is not a single agency. It is not the only single agency, but it is a collaborated, coordinated effort of all the disciplines from all fields of science. So hence it is called a multidisciplinary approach. Now what is the significance? What is the importance? Why should we involve multidisciplinary expertise? Now the reason is that in order to understand a comprehensive analysis, that means you have to give a holistic kind of a analysis, and then it has to be very, very accurate. When you give it to the proper expert, only then he will be able to give you the accurate conclusion. So for these two reasons, then we may have to use the multidisciplinary approach whenever we talk of the forensic expertise. Now let us move on to the challenges. Next slide is talking about the challenges in the forensic molecular biology. Now what are the challenges? As I told you, contamination is the biggest risk. Now if we don't adopt the standard operating procedures, either in handling, either in lifting or transportation or storage, any one of these activities, if you are not going to take the right kind of activity, what is going to be the end result? It is the contamination. Now in order to prevent this, follow the guidelines that are given very, very scrupulously, is what is about contamination. Now degradation. Certain kind of a degradation is inevitable. It is not in your hand. Environment you can't change. Adverse temperature you can't change. This is all in nature, you can't control that. Now what is the best way? Only way is, if a particular material is prone to degradation, ensure such a methodology where the degradation is minimal, or the degradation can be arrested, isn't it? That degradation cannot be compromised. So adopt such methodology. Now, what are the ethical issues? Privacy concerns. Now, and consent. For example, I am an accused. Now, I have got lot of immunity. Just because I am an accused, that everybody can interrogate me, everybody can question me, everybody can arrest me without giving the reasons, everybody can prosecute me. No, I have got my own immunities. Now my immunity is to remain silent. If somebody asks me some question, I can remain silent. If I know that the answering of which is going to incriminate me, I can resist. I can say, I have this immunity, I have privacy concern. Now somebody wants to take my blood. If I don't give consent, nobody can force me to give blood, isn't it? So these are privacy concerns, and then lot of constitutional privileges have been given to us under Article 23 and then Article 21, 20, lot of privileges and privacy things are given to the accused. So that is the reason why we say, whenever we try to take some samples, especially in cases like narco analysis and then the polygraph and all, when you want to study the psychological profile of the accused, then it is but natural that you have to take the consent, that is written consent which is not given under force. So the moment that the defense can prove that, see this people, they have taken my consent, putting me under pressure, putting me under fear, because police are experts in doing that. So if the accused or from the side of the accused, if the defense can prove that, then that consent is no consent at all under the eye of law. So ethical concerns also have to be looked into. So there are also major challenges in the field of molecular biology. So moving on to the analysis part of DNA analysis. How much time the organizers can tell me how much time still do I have? What is the time allocated? What time up to?
45 minutes left.
45 minutes left. So moving on to the DNA analysis per se now. Please change the slide. Move on to the slide, next slide. DNA analysis, then also we need to understand what do we mean by the DNA analysis, and then what are all the different techniques that are available to the lab personnel so that he can perform the DNA. And there is an evolution from the day one which has been the DNA has been, technology has been discovered till date. There are a lot of changes that have taken place, and then we have the RPLF technology, Amplicon technology. So there is a great evolution in the adoption of the technologies, and some technologies, to name few of them, then we have polymerase chain reactions, short tandem repeats, mitochondrial DNA analysis. So there are a number of analyses that are available, and depending upon the material that is there in hand, we may have to adopt the methodology. Let us move on to the other slide. Please move to the other slide. Now, when we say analysis is not a single step process, it is a multi-step process, and we need to understand what are all the steps that are involved. Now, the first step is extraction, the second step is quantification, third is amplification, fourth is separation, and last will be the analysis. Now, what do you mean by extraction? Now, we know that our human system is provided with a lot of safeguards. So our skin, we have the internal organs, delicate organs, our tissues, our bones are all properly being covered by a skin, isn't it? And skin also, inside you have tissues. Tissues are composed of cells, isn't it? Cell is not bare, it is protected. There is a cell wall on that. Now inside the cell you have nucleus, inside that you have chromosomes. The entire thing is called a nucleus. In that you have got a DNA and all. So in order to understand the DNA, in the first place DNA has to be taken out. isn't it? That has to be the first step. So that is what exactly we are going to do in the first step, that is extraction. Whatever DNA that is available within the cell, you have to open the cell wall, you have to break open the nuclear membrane. From there you are going to extract the DNA. So this process of taking out DNA from the nucleus, from the cell, is called the extraction process. Now isolating DNA from the biological material, for example it is blood. Now blood is a complex tissue, again a connective tissue, fluid connective tissue, which is composed of different cells, RBC, WBC, platelets and all. But one point each student should understand is the red blood cells are otherwise called in scientific terms erythrocytes. Erythrocytes of human beings of adult category, adult matured RBC, are devoid of nucleus. You understood what I mean by that? Mature human RBC are devoid of nucleus. That means they don't contain nucleus. So they are unfit for DNA. Then anything which contains DNA, anything which contains a cell, single cell, that is that is the minimum criteria that is required. So I am saying that in the blood, RBC are without nucleus, they are devoid of nucleus, hence they are unfit for DNA. Now, why are we still banking upon blood? Blood is the best source. Anyway, in any crime, blood is bound to be there. Now, if RBC are devoid of nucleus, then which other tissue in the blood should we focus? Which other type of cell should we focus for DNA then? I told you RBC are devoid. Now, what are the other two types of cells in the blood? Yes? What are the other two cell components apart from RBC? Platelets and white blood cells, isn't it? So white blood cells are relatively bigger and they do have nucleus. So that is the voice is not clear. Is voice clear now? Louder?
Yes, ma'am, now clear.
Now clear. So what I am trying to ask you is, now I said human RBC matured are devoid of nucleus. That is the statement I made. So what do I mean by that? When once a cell which is destined to become a human RBC, that matured thing will not have any nucleus. That means it is unfit for DNA. Minimum that is required for DNA is a nucleus and then cell. One single cell at least minimum is required. So the RBC do not have any nucleus at all. So they are unfit. Now what is the other cell that we can depend upon in blood? That is WBC. So WBC in blood is the only cell from which you can extract the DNA. Now quantification, because we require a minimum amount of DNA to proceed further in DNA analysis. Now the next step after extraction will be quantify. In the given sample of evidence, how much DNA is present? Most of the time in forensic samples, it is always the minimum sample is available. Good DNA in the first place is very difficult because there are so many factors trying to destroy the material. So even if some good sample is there, the quantity is not sufficient. Why? Because only some minimal quantity is required for you to process in the DNA analysis. That is not there in our given sample. So should we stop there or should we go ahead? So what is the step that we have to take? Now there are techniques available. Whatever quantity that is given, that is there in the given sample, we can amplify it, we can multiplicate it exponentially. Now in PCR and all, there are techniques where we can amplify, we can magnify, we can increase the copies of the DNA. So this is the step where you will exactly find out whether the extracted DNA, is it sufficient in quantity to proceed further, or does it require a step which will amplify the quantity of the DNA, is what is known as the quantification step. Now next is the amplification step. As I told you, it is going to a technique where you are going to generate copies exponentially. This is called polymerase chain reaction, in short PCR, polymerase chain reaction. Now after this third step of amplification, where you ensure that I have run the PCR, now I am confident a minimum quantity of DNA is available in the sample now, I'll go further, I'll go a step forward, and I'll go for the separation step. So what exactly are we going to do? In the separation, the DNA that is there, the DNA quantity, enough quantity that is obtained at the end of the amplification, you are going to subject it to a chemical seizure, that is endonucleases, which is going to cut the DNA, the required in at specific sites. Now the result, you are going to get some fragments of different sizes. So these fragments need to be separated, they need to be segregated, and by fragment size, by length of the fragment that are the end products of endonuclease, as per the size of the fragment, you are going to separate the DNA fragment. And finally, once all these fragments are segregated as to the size of the fragment, then the final step will be the analysis. So when I say analysis, if suppose there is a case of, let us say, the parentage dispute, a child is there and the mother claims that it is the child of a particular gentleman whom she claims as the father of that child, and that gentleman is going to deny, I have not fathered the child, he is going to deny that. Now, is profiling of a child DNA alone is sufficient? You need to have some reference samples against which you are going to compare. After all, analysis is a comparison process. Now, in this particular example of paternity case, what are all the samples that you require? You require the blood sample of the father, so-called putative father, whose parentage is in dispute, then the mother's blood and the child's blood. So, all the three samples of blood are required. Now, you are going to extract the DNA out of the child's blood, you are going to extract the DNA from the father's blood, you are going to extract the DNA from the mother's blood. Since it is a case of dispute, now the child's DNA is compared against the father and mother. Now, as per the principles of Mendelian, then child has to inherit whatever there in the father and mother. So, if a particular gene is not present in the child, which can be attributed to the father, definitely that he is not the father. That is how the Mendelian law works. Now, as per that, you are going to compare the DNA profiles of the child against the father, against the mother, and then you are going to assess and then say whether the child belongs to the given parentage or not is the technique. Now, DNA technology and profiling. Let us move on to the next slide and try to understand after these steps then what exactly is the DNA technology is all about and what is the profiling is all about. Now, in that definition, if you see, using DNA to create a profile unique to an individual. Now, we know fingerprints are so unique, normal fingerprints are unique, and they are going to be there throughout, and they are not going to be changed with age or with deformity. That is what we understand by DNA. That is what we understand by the fingerprints. Similar principle can also be applied to the DNA. Once DNA of an individual is established, it doesn't change. Whether it doesn't change as per the source, whether it is blood, whether it is bone, or whether it is teeth, or whether it is skin, any part of the same individual, as long as it is coming from the same individual, it is going to be unique. It is going to be its exclusive profile only. Now, that is what we mean, because each individual has got their own unique, separate kind of a profiling which cannot be duplicated by anybody else. Now, that is the definition, and as I told you, as of now, there are many technologies that are available, latest being the STR analysis, restrictive fragment length polymorphism is one, single nucleotide polymorphism is another technology. So, there are many technologies. Again, which technology has to be used will be dictated by the sample that you have in hand, how much sample is available, how much good DNA is available, whether you are dealing a case of mixed sample like in rape case or gang rape case. So whether it is a POXO case, depending upon the case that you are dealing, the type of material that you are dealing, the technology will also accordingly differ. Now, what is the application? Why do we use technologies, DNA technology in forensic field? Because forensic deals with the crimes, in resolving the crimes, And whenever criminal identification, because in any crime investigation, two important aspects are only there. One is to establish whether the crime has taken place as per the definition of IPC. Second, who has done it, who is the perpetrator of the crime. To understand who the perpetrator of the crime is, you have to identify the criminal. So, criminal identification, DNA is the best. In order to understand who the murderer is, you have to do the profiling of the murderer's blood. In order to know who the rapist is, you have to profile the rapist blood for DNA profiling. So, in order to know who the person who has committed the POCSO case, then similarly you have to profile that person's DNA. So, accordingly, whenever identification of the criminal is involved, it can be a paternity case, it can be identification of the remnants. Remnants when you talk of disasters. So, disaster, as I told you, it may not be possible every time by looking at the physical features. Even kith and kin relatives, most of the time there were erroneous identifications, misidentifications. With the result, they were carrying wrong bodies, and then there were many problems. So there also identification is a biggest issue. And unidentified bodies, there may be claimant for that. They say it is our body, then maybe with DNA it may prove the other way. It may not be their relative kith and kin. So wherever such disputes or ambiguities are there, it is but natural that we need to go for the identification of DNA processes. Then, moving on to the other slide. Let's move on. Please change the slide. Now, as I told you, there are several steps again. Now, what are all the steps we need to understand? Collection, storage, characterization, we have understood this. And here, there is a blood stain. In the example, you can see that there is a blood stain. It can be a buccal swab. So, blood stain, the origin is not known. Now, you are suspecting somebody, you are asking them to take a buccal swab. Cheek swabbing is taken. Now, sample is properly stored, and now it is been sent to the laboratory. Now, in the laboratory, you try to extract the DNA. You have also quantified the DNA from both the samples. What are the two samples here? The blood stain that is available in the scene, and the cheek sample or the buccal swab sample you have collected from the suspect. So these two samples are there. You need to compare the DNA profiles of these two and then say whether the origin of the blood that is present in the scene, has it come from the suspect whose buccal swab we have taken. So in order to do that comparison, we need two samples. Now in the lab, what are we doing? We are trying to extract the DNA from both these samples, and when we feel that the quantity is little less, we are going to do the PCR quantification, after quantification going for PCR, and then we are going to do the STR marker analysis for that. So then after that, for interpretation and for separation and all, we are going to generate lot of data, and there are capillaries, electrophoresis and all. These are detection techniques. Now with the help of these detection techniques, we are going to get some electrophoreograms, and these electrophoreograms, there are peaks, and each peak is identified. There are softwares available with the gene mapper software. So it is going to give you peaks with numbers. Then you have to see, mix and match, see the same type of STRs, whether they are available in mother, in father, if it is a paternity case. In this case, if the DNA samples from the blood stain and the DNA from the buccal swab of the suspect, if they are exactly matching, if they are superimposed one on the other, that means the source of blood that is present in the scene has come from the buccal swab DNA. Since these two are matching, we can say the person is the same. The person suspect is the one whose blood is dropped at the scene of offence, is the conclusion that can be drawn. If you see up to this capillary electrophoresis, this is called the measurement aspect, and when it comes to the data interpretation, the electrophoregram and all, statistical techniques and all, this constitute what is known as the interpretation. So that means there are two parts in the FSL, the forensic science laboratory. One is you measure, you identify, You measure, you extract the DNA, and then a profile is being generated. Once the profile is generated, it is the interpretation. It is the interpretation means how you are going to give a report on that interpretation of that data from the electrophotogram, and then you are going to say explicitly whether the child belongs to the given parentage in case of a paternity, or in case of a murder, whether the blood stain that is there on the weapon and the blood stain that is there on the scene, whether they are one and the same. So like that you are going to give the interpretation. So last step will be the interpretation. Now there are STR markers at the bottom I have given. There are different markers. Markers are spread all over the genome, and the length of this marker is what at present we are going to do, whether it is 7 number, 8 number, and individuals the number of repeats will be varying, and in mother and father it is combination of egg and sperm. So how many repeats are coming from the father, how many fragment length are coming from the mother, father. So based on the child is going to get it at random in a permutation and combination. Even the siblings of the same parents, you don't see that there is a repetition. So that is the reason we said the STR markers are per se the DNA is so unique that you can identify. So underneath that I have mentioned some of the equipment for PCR, genetic analyzer, where the capillary electrophoresis is used, and there is a software which is going to give you the electrophotogram, and then you are going to interpret the results. So this is these are the steps that are involved in the what you call the analysis aspect. Now moving on to the other slide, which talks about again the collection, analysis, comparison. Move on to the other slide. Am I clear so far? Whatever I am teaching, I am making sense, I suppose. Is it so clear? We are coming to the fag end of the lecture.
Yes, ma'am.
Huh.
Yes, ma'am.
That's it. Then this slide talks about now what do we mean by collection, what do we mean analysis, and then comparison. As I told you, collection is always, for DNA samples, you need to collect the biological. It can be a swab, it can be a buccal swab, it can be a anal swab. In case of Oxford, it can be a anal swab, or it can be a cervical swab, isn't it? Or it is a swab from the pubic region. So you have to collect from different regions the samples, and from there you have to extract the DNA. Now analysis part, where you are going to extract the DNA, and if you feel the quantity is less, you are going to amplify the DNA, and once the required quantity of DNA is there, then you are also have the samples, reference samples, comparison samples. So per se, any profile will not give any information. Only when you compare it with the reference, if there is some matching points. Like in fingerprints, how do we do normal fingerprinting? Fingerprint from the scene of offense and fingerprint of the accused. If these two are matching, we say that he is the person who is involved. So similarly for DNA also profiling, you need to have the reference samples, known samples, whose DNA is known to us, whose source is very much known to us. So moving on to the other slide, which talks about the human DNA genome within the cell. Next slide, please. So here we are talking about two different DNAs. One DNA is available in the mitochondrial DNA, which has got a maternal lineage, and one DNA is there in every human being, that is nuclear DNA. And there are quite number of differences between these two DNAs. One is circular, the other one is helical, and one is maternal lineage, and the other one is universal. And so like that, there are, a few copies are there for nuclear. One per cell is the limitation in nuclear DNA, unlike the mitochondrial, which is the powerhouse of the cell, and this is the center of the cell when you talk of nuclear DNA. Now there are a number of contrasting points. You see the base pairs now, mitochondria being less base pair, whereas when you talk of nuclear DNA, it has got more base pairs. And similarly, as I said, the lineage as the inheritance, if you see mitochondrial DNA, it has got a maternal lineage, and whereas the nuclear DNA, if you see, it is inherited both from father and the mother. Now this is about the human genome within the cell. Moving on to the other slide. which talks about the genetic inheritance, how the genes are inherited from father to mother or from parents to the offspring is depicted in the next slide. Move to the next slide, please. Yes. So this is talking about the genetic inheritance. Now, whenever all human beings are they are called in their initial stages zygote, which is a result of fusion of father's sperm and then mother's egg. So these are uniting, and then at the end of the generation, the number of chromosome component is maintained, and initially the father's sperm will have a haploid number of chromosomes. That means all human beings, we have 23 pairs. So why we are calling it pairs? Because each chromosome has got maternal origin and then also one from the father, paternal origin. That is how we have a double component of the 23 pair. Now that means the father's sperm should be haploid, it should contain only one chromosome, then hence it is called 23 single haploid chromosomes. And similarly, egg of mother also has got a single component, that is called the haploid number of chromosomes. And when these two fuse, that means when the egg of mother and father's sperm, when they fuse, then it results in the formation of a child. Initially it is called zygote. Zygote, as it develops into a child, and that's how the inheritance happen. You can see the process of inheritance in a child. Now we know that there are autosomes, and which don't determine the sex of the person, because male and female child is genetically determined, unlike the myth that lady is responsible for male child, female child. These are all myths. Under genetics, it is the father who is going to determine the sex of the person or the individual. Now father contribution is 22 autosomal out of 23 chromosome component. Father is contributing 22 autosomes, which are not going to determine the sex, and only the last pair is going to determine. That means the X chromosome is present both in mother and father, and if it is in double dose, that means father also contributing X chromosome, mother also contributing X, so it becomes a homozygous pair XX, and that determines the sex, and the resultant will be female child. And instead of X chromosome, apart from the 22 autosomes, when the father, instead of X, when he is contributing a sperm which has got a Y chromosome, then 22 autosomes plus one Y chromosome, that results in the formation of a female male child, because one X is coming from the mother and one is coming from the father in the form of Y. So resultant will be XY, and that gives to the formation of a male child. So double X will be female, single X with Y will be male child. So that means in human beings, it is the father who is going to determine the sex of the child. Moving on to the other slide. So if we are running short, or if we have completed the time, it's only just giving more inputs. So if the time is up, you just prompt me. The organizers have to prompt me so that I can just stop and then take a couple of questions. Are we still have time? Do we have time? Can I go ahead or take questions? What do you feel? It's an unending kind of a subject. I can go on like that.
We take 15 minutes and
We'll go for 15 minutes and then take some questions. That should be okay? So you should, because in the flow I may not be noticing the time, you should prompt me wherever I have to stop. Whenever I have to stop, you have to tell me, you have to prompt me. Okay? Now moving on to the, this is the slide where human genome, how it is inherited is given. Now if you see, there is one egg, it is written, then sperm, then bottom it is written as the zygote. So as I said, the egg from the mother, when it gets matured, and the sperm from the father, they are going to unite, and this is what the upper portion is showing, the autosomes. 1 to 22 are only autosomes. They are not going to decide the sex of the person. So from the mother, in haploid thing, in single chromosomes, they are being contributed from the mother, and then she is going to contribute one X. She can only contribute only X. She doesn't have any Y. So mother is going to contribute only X, and then there are also mitochondrial DNA in her in her cell. Then these two are combining. You can see the sperm, which is also haploid, which has got only a single chromosome, and from 1 to 22 labeled, you have autosomes, 22 autosomes. They are not going to decide the sex of the person, and only the sex chromosomes. Unlike the mother chromosome composition, the father's composition has got a X and Y, both chromosomes. When he is going to contribute only X, for X of mother, the resultant will be double X, and it will be a female child. So in other combination is the along with the autosomes 22, if he is going to contribute not X but Y, so it will be a combination of X from the mother and Y from the father. So the resultant child will be a male child. This is how the sex of the person. In the zygote you can see it is represented double line, whereas in egg it is only a single line, and in sperm also it is a single line. 1 and 1 here, both are represented single. So 1 and 1 will become again a double 1 here. Zygote, if you see, there is a double line that is written. So that means it will be combined. The chromosome 1 is inherited from mother, and 1 invariably has to be from the father. So this is how male or female child, at bottom it will give the full component, full genome of the male child. And of course male child do not inherit the mitochondrial DNA. It is only the mother's lineage that is present. Only mother, that means from maternal aunt it will go to the child, female child, and from the grandmother it goes to the mother, from mother to the offspring. Like that, only the maternal lineage will be there with regard to the mitochondrial thing. Likewise, Y chromosome lineage also will be exclusively for the male child. This is what you need to understand from this particular slide. Moving on to the other slide, which talks about STR. Yes, move on to the other slide, please. Please move on to the other slide. Is there anybody to change the slide? Please move on to that. Yes. STR is what is we have already tried to define. It is called short tandem repeat. Tandem means side by side. Short, it is short because the length is short, and then repeat means it is going to repeat throughout the length of the genome. That's what you need to understand. In STR technology, what we try to do is tag the ends of the portion which we are going to analyze, that particular STR region, a particular region which is composed of, let us say its length is 7. Now both the sides of this region they are called the flanking regions. We are going to use some dyes which will become visible in the examination. We are going to tag the fluorescent dyes, primers on both the sides, and it is going to happen on both the sides. You can see the arrow which is called forward PCR primer. The reverse is called the reverse PCR primer. You are using this technology, and then currently we have only this length polymorphism is being measured. Now you see that these repeats, there may be 7 repeats, there may be 8 repeats, or they may be that means in a particular child, he may inherit 4 repeats from the father, he may receive 5 repeats from the mother. So the component of the STR length keeps changing, and it is the combination, permutation of these repeats from father and mother that will be going to the siblings. So that is how you are going to analyze how many repeats are there, and which repeat has come from father, which length has come from the mother. Based on that you are going to profile the DNA of the child. Now as of now, is there any restriction? How many STRs? Because n number of STRs are there throughout the genome, I told you. Now our concentration should be only on one particular region which is variable, because we know the entire DNA is going to be the same, whether it is for chimpanzee or whether it is for human or whether it is for anybody, it is going to be the same. But then only that 0.1 will be varying, because every human being or every living being will have the same digestion system, will have the same digestive juices and all. The variation cannot happen in these things. So variation can happen only in that 0.1 portion of the DNA. So that is called how many identified STRs so far. So in FBI originally I selected only 13. Now currently there are commercial kits which are available, which will go as many as 20 to 22 STR loci have been identified, and they are going to be interpreted. Now moving on to the other slide. which talks about autosomal DNA results from a single individual, two different loci. Now you see this is very, very interesting. Now examine very closely the figure, schematic figure that is given. In 140, 180 region, if you see, there is a long peak, and the nomenclature of it is 14, 148.41, 5584. So what you can see is only a single peak, which is a very big peak that is being given. Now when the STRs from the mother and father, when they are the same, when STR alleles are the same, that is from the father and mother, it will be given as only a single peak. That means it is homozygous. Homo means same, hetero means different. So a single large peak in the electrophotogram, if you are getting only a single peak in the child profile, that means father and mother, the STR alleles that are contributed are the same. They are merging, they are appearing as a single bigger peak. That's what you have to understand. Unlike the profile that is given on the extreme right side, if you see, there are two small peaks that are represented. Are you able to appreciate those two peaks? Unlike the peak which is bigger, where it is homozygous, in heterozygous you are able to see two different peaks which are smaller. Are you able to appreciate that particular fact? Can you see two different peaks there?
Yes, ma'am.
These two are different and they are shorter. What does that mean? They have different origins. That means the STR from mom and the STR from dad, they are two different. Two different in what? In what aspect? Lengthwise they are different. We have seen 1 to 7. There are different lengths, isn't it? So when the STR length that is being contributed from mother and father, if they are of different sizes, you are going to get two different peaks. So it is after all only the peak. In the electrophotogram, what you see is only the peaks. So from the peaks you are able to interpret. What is the interpretation that you are able to make? Suppose if it is in the electrophotogram only one peak, very big peak appearing, then the conclusion that can be drawn is the STR contribution from the mother and father is the same. The allele is the same. That is the reason there is a merger of alleles and then you see a single peak, very big peak. But in contrast, if the length of STR alleles from mother and father, if they are different, then you are going to get two different discrete kind of peaks. That means they are heterozygous for that particular STR allele. Am I clear about this? Yes. Move on to the other slide. I think this will be the last slide. I will be showing the rest of the slides. I think I need not show. It is better that I take some couple of questions so that whatever is being taught, it should be first understood. The slides can go on, explanation can go on, but there is no point if somebody has not understood that, and then mere going on and mere explanation will not help unless the other side also understands what I am saying. So this is simply, if you see the 13 to 20 loci that are there, there is a nomenclature for understanding. Each loci is given a distinct name. You see D1, S1, 656. There is some kind of a nomenclature that is given, if you can understand. Now, so there is a different nomenclature. I think perhaps next slide will be the last slide. I will tell why this D and what S stands for, what 317 stands for. So there is a nomenclature. I will try to just explain the locus name. Then I think we can stop after that slide. Yes? Let's move on to the last slide. Now next slide will be the last slide, I said. Yes, move on to the next slide. Am I audible? Please move on to the next slide. Or do you think that we should?
Yes, ma'am, you are audible, but we have issue.
See, this will be the last slide. There are still slides remaining. I don't want to proceed further, but then I'll stop after explaining this particular slide and take a couple of questions. That will be good. Now, when we say D1, 3S, 317. There is one nomenclature, one name, like we have surname, we have proper name, father name and all, like that even the STRs are given some name. Now let us try to understand this. D means DNA, that is understandable. 13 means chromosome 13. S stands for a specific sequence, and 317 means the position of that particular allele or the particular locus in the entire genome is 317th location or position is what is. So similarly other kind of alleles or STRs are given different nomenclatures. I think I should stop at this point, and then you can stop sharing the screen, and I would like to take a couple of questions if somebody has got questions.
Thank you so much, ma'am. Any question?
You can be
Question answer session?
yes. Those who want to ask questions can be little closer to the mic so that they are more clear and they are quite audible.
Ma'am, my question is that how long can we keep DNA samples preserved?
See, if you follow the standard operating procedure, lot of manuals have been written, lot of labs have been accredited, they maintain quality standards and all. You scrupulously follow what is given in the manual and maintain the cleanliness of the lab, and you can preserve it for very long. And the best way to preserve is, don't preserve it in the form of a biological sample. Now the moment, because most of the time what happens in the resolving a case, you go to the scene of offense, you have the sample ready-made available. But then when it comes to perpetrator, it takes time. Investigation, most of the time, identifying the perpetrator who has done the crime takes lot of time. But you can't keep waiting for that, Meanwhile, what the IO has to do is send the sample as it is available in the scene. So whose DNA is not known. But then if you send it immediately, if the IO sends it immediately to the lab, what he does is, he will isolate DNA from that sample, keep it in the refrigerator, defreeze it for long. Once it is extracted and kept in deep freezer, it is going to stay for many more years. And when in the investigation, if the IO can identify the perpetrator at a later time, he is also going to send his samples also. Now the lab, what it does is, they have already crime sample, isn't it? Now the reference sample is also being sent by the IO. They are going to extract DNA also from the reference samples. Now they are going to make a comparison between the sample, between the DNA which they have extracted from the crime sample, and now from the reference also they have collected, they have generated the profile, DNA profile. So they are matching. If they match, it is the accused blood that is being shed in the crime scene, is the conclusion that can be drawn. So again it depends. Don't wait till the identification of the perpetrator. As and when this is detected, send it across, and once the DNA has been extracted, once the isolated DNA is kept in deep freezer minus 80 degrees, it is going to be there for very long period. Or DNA, which is the biological sample collected on FTA cards. These days FTA cards are available, company made cards which have got lysed preservative there. If you collect blood on that, they are going to be preserved for a very long time. So you have to follow what is written in the manual, standard operating procedures. So it will remain forever.
Thank you.
I hope I have made the answer quite clear. Yes, anybody else?
Yes, ma'am. Am I audible?
You are audible. Bolo. You can tell your name and ask the question, so that I will also understand that the person who is asking is so and so.
Ma'am, my name is Rishna Gupta. Ma'am, my question is, rather than human DNA, if we got any kind of animal DNA at the scene of crime, is it the same procedure to preserve it or to collect it?
Same procedure will be there for animal blood also, but only thing is, different technologies have to be used. You cannot use the same probe which you use for humans, same thing for animals. There will be different technologies, different, as far as the procedure of collection of the samples and all is concerned, same procedures have to be followed. But when it comes to analysis in the lab, probes and technologies will be different. For animals, you may have to use the population, how many alleles are available, whether there is a diversification in the alleles and all. There is a population genetics which talks about all this. And then a particular gene, it has to be frequent and it has to, it should have an equal distribution among population. And so many factors are there. When you talk of animal DNA, you should also study the population genetics of the animal population that are residing in that particular geographical location. Probes will be different, but procedure for collection of the biological samples, as far as that aspect is concerned, it is going to be the same for human and animal equally. Am I clear?
Okay, ma'am. Thank you so much.
Yes.
Yes, ma'am, I do have one question.
Yeah, please go on.
Yes, ma'am, my name is Pratiksha Patika.
Huh.
Ma'am, I want to ask that in twin case, how we compare the DNA? If we get any type of case and there is a twins, then how can we compare that? How can we get the exact one?
And there are techniques, SNP techniques, as I said, while I was discussing, I said RFLP, STR, like that SNP technology, single nucleotide polymorphism. So that type of techniques are taking us to such an extent that even identical twins, identical monozygotic twins, they also can be identified. Technologies are not very far where we can also go a step ahead than identify the twins, differentiate between the identical twins or monozygotic twins also. Technologies are, they are available. Earlier it was difficult, but then with new technologies coming up like SNP and all, we are able to do that. Yes.
Okay, thank you, sir.
Yes. Any other questions? Yes. Are there any questions in the chat? Somebody left any questions in the chat box? See, students have to be vocal in such forums, otherwise if you keep your doubts for yourselves, that doubt will never get clarified. Because you are not visible, you need not feel shy. You can close your video and then ask. If you want to be anonymous, you can do that also. Point that is important is that your doubt should be clear. That is the thing. Yes, anybody? If there are no questions, I think we can leave the meeting.
Yes, ma'am. Yes, ma'am. So thank you so much.
Age of an individual from DNA. I am afraid there are, as of now, I don't think there is any technology which can differentiate which can tell us when the DNA is deposited, how many hours or days before it is been shed and all. I mean, these are, but nevertheless you can identify the phenotype, not only the genotype. With the DNA technologies, phenotyping is also now possible. Age, maybe future days to come will be successful in determining the age of the person also. Phenotype now, yes, possible. But the age, I am doubtful. As of now, we don't have any technology to tell the age of the person.
I would like to invite Anamika ma'am for vote of thanks.
Good afternoon, ma'am. It was indeed a very nice session, and it was some very nice insights about the policies. And I'm hopeful that really have more such sessions with you. Thank you so much, ma'am, on behalf of the Institute of Sciences, State University.
So it's also a great platform whenever I interact with students, because I'm almost 67-year-old. So whenever I interact with people, young people, I feel my age is getting drastically reduced to your level. So that way I feel, the more I try to interact, in the process I am getting educated. That's how I take it, and it's a wonderful time with all of you. And hopefully that all your students will be well placed in their jobs, and then they are going to have a very good future, because forensic as such has got a very bright future in days to come, thanks to the Naya Kanun, where it specifies that a lot of scope for the forensic scientists, and let us hope for the best. Good luck to everybody.
Thanks, ma'am.
Thank you, ma'am. Thank you, ma'am.
Yes.
Thank you so much, dear audience.
So we'll leave the meeting now.